In part 1, we examined Eugene's ultra deep subsurface biosphere. In part 2, we will look at the evidence that underpins this model and discuss some of the implications that this might have for Earth and extraterrestrial life. So let's explore the evidence for Eugene's ultra deep biosphere. So what evidence is there to date to support this hypothesis? Let's start by examining methane emissions. One possible sign of microbial activity would be the emissions of biogenic methane at or around earthquake zones or fault lines. The release of methane at these places has been observed, for example, at the 2010 earthquake in Chile, the Macran Trench in 1945, and outventing of methane gas at times of relative calm along the Cascadia Fault. Eugene points out that although some of these studies have attempted to differentiate between biogenic and non-biogenic sources of methane based on an isotope of carbon, he points out that this may not be a good method, as laboratory tests have shown that certain microorganisms can change the ratio of this carbon isotope depending on the environmental conditions. Now, it is well known that fracking causes earthquakes. There are two ways in which this can occur. The first is via the fracking itself, injections of fluid into the hydrocarbon-bearing rock, and these are rarer and cause weaker earthquakes. The second more common way is through the disposal of wastewater via the injection into the deep storage wells. If we examine the second way, it becomes obvious that as you inject salt water, this could easily provide a response in metabolism of these organisms. Not only does it provide them with water itself, but it is highly conductive, which may play a significant role in the enhancement of extracellular electron transport processes, or telluric currents. We know that volcanic activity is related to seismicity, so the ultra-deep biosphere might also play a role in these processes as well. This is supported by contemporary models which show that the depth of volcanic magma reservoirs is only at about 10 kilometers, which could easily be accessible to these microorganisms. The only problem is obviously the high temperatures involved, but there are many examples of microorganisms that are found around volcanic rocks and lava flows. In the recent Icelandic eruptions, samples of lava were shown to contain a species of microorganisms that did not require light, potentially indicating that they did not appear into the cooling lava, but were present from before the eruption. He goes on to cite many examples of sudden changes in the local microbial community, around underwater rifts and volcanoes, clear evidence of a sudden influx of new microbial life, potentially associated with the rift or the volcano. A consequence of this concept is that these microorganisms might end up being released closer to the surface into other ecosystems. They might be pathogens themselves, or they may modify other microorganisms through horizontal gene transfer, which might pathologize them. So is there any correlation between post-earthquake infections? Now, clearly earthquakes themselves may cause massive devastation and cause problems for sanitation and overcrowding in shelters. There is, however, evidence of changes in certain pathogens after earthquake. A fungus, Candida auris, simultaneously emerged on 3 different continents from 2012 to 2015. Most notable is that they had a much higher tolerance for elevated temperatures. Could this have been caused by the Japanese earthquake in 2011? Another example is the earthquake in Haiti in 2010. This was followed by a catastrophic cholera outbreak. Before this, cholera had never been observed on the island, although initial findings suggested that this was probably brought in by the United Nations workers. A more recent study indicates that the biotype was different and may indeed indicate a different origin. In addition to the many points we discussed on adaptation, it is important to consider that some of these microorganisms may have developed additional tools to aid in their survival. The first is to consider that they may have developed some type of protective shell. There are indeed examples of other microorganisms doing just this, like the nostoc bacteria colonies, which form an extracellular matrix and might reach 0.17 mm in size. There are even colonies of algae which are able to produce durable silicate scales and spines for protection. Rotifers are an example of a microorganism which is known to grow a protective exoskeleton. Another possibility is the formation of a biofilm that enhances the protection and could make the recycling of surrounding materials easier. We must also consider that different types of microorganisms might produce different specializations and could work together in a symbiotic relationship. This behavior is found in many unicellular organisms. An example is the Stenta polymorphus, which keeps chlorella algae inside its cells to provide protection for them, and receives maltose in exchange. Another possibility is that the microorganisms can shut down their metabolism in order to protect themselves from the harsh external conditions. Tardigrades produce a protein that allows them to vitrify themselves to help them survive during severe dehydration. This means that these microorganisms remain dormant for long periods of time and then spring to life as the conditions become more favorable and trigger an earthquake. An extreme example of this are bacterial endospores. Here, microorganisms are able to sense and adapt to the changes in the environment. When the nutrient source is exhausted, some bacteria can employ an extreme survival strategy. They undergo a process of change and produce a cell which is dormant and highly resistant in order to preserve the cell's genetic material. Studies have shown that these cells can survive an unbelievable 10 to 100 million years. Dormant endospores are present in marine sediments worldwide. Eugene goes on to suggest that some more extreme adaptations based on the phase transition of water postulated by Pollock might also be possible. He also suggests that they may have adapted to radiate heat quicker, reducing their overall thermal load. This could be achieved by the synthesis of a material similar to Vanta black, which could be created at temperatures accessible to life. Another possibility is that rather than radiating this heat away, they could utilize it as a source of energy. If they are able to form long chains and perform distributed electron transport, they might be able to use the temperature gradient in the crust to create a sort of biological thermocouple to drive the metabolic process of life. The formation of hydrocarbons is thought to be a slow process, and it is assumed that the creatures die on the surface and then need to be buried under many layers of sediment, which causes an increase in pressure and that causes the formation. This hypothesis might provide an alternative mechanism for the creation of hydrocarbons at extreme pressures and temperatures and allow for a much more rapid production of them. This could help explain why there have been hydrocarbons discovered in what is considered geologically young formations, which cannot really be explained by the present models. One hint towards this concept is the similarity between bacteria found in warm subsurface petroleum reservoirs and bacteria in the oceanic crust. So it may be possible that these organisms are responsible for both the production of methane and the synthesis of more complex organic molecules. As this concept assumes that choleric currents might play an essential role in the biogenic earthquake production, it is reasonable to consider that other key electrical systems of the planet, like the global electric circuit, might also be important. It is considered to be limited to just the atmosphere of the Earth, and the role of the underlying areas are seldom discussed. Eugene starts by tackling the atmospheric circuit. Recent studies have indicated a significant impact of the microorganisms on sea spray aerosol properties. There may be a link to the evaporation process, which in turn imply this could be modulated, which would directly impact thunderstorm activities, ionospheric potential, and the vertical electric current density in the atmosphere. They have even conducted experiments to see if it is possible to transfer microorganisms from the ocean to the atmosphere via this process, and they discovered that by enhancing the cell membrane to have a hydrophobic envelope, it increased the changes of aerolization. Some decades ago, the concept of bioprecipitation was introduced. This was the idea that biological organisms might noticeably enhance cloud condensation and the related phenomena. It is known that the biological ice nucleators are always the most active, and thus some bacteria might promote quick ice nucleation in the atmosphere. They have been found in snowfalls all across the world. The bacterial proteins they produce are actually used to create artificial snow. Again, this goes to show how underestimated the role of microorganisms is in the atmospheric chemistry, global weather, and climatic patterns. It has recently been shown that the water microdroplets spontaneously lose electrons, producing hydrogen peroxide. Eugene postulates that this process might be used by aerobic bacteria in the atmosphere for energy acquisition. The mysterious noctilucent clouds might be explained by the presence of these microorganisms in the atmosphere. Eugene goes on to suggest that the ultra deep biosphere might also be at least partly responsible for the electrical polarization of the crust. Conventional models certainly show how the potential difference between the crust and the ionosphere can be maintained, yet the question of generation of this potential difference still remains open. If the microorganisms in the ultra-deep environment possess the same degree of negative electrical potential as any regular cell, it would induce positive charges on the interfaces in the immediate vicinity, which would, in turn, negatively polarize the surface of the crust, so the correct charge sign would still be observed. It is known that the propagation of positive charges from the underground depth to the surface increasing air ionization. This in turn can lead to various atmospheric phenomena such as earthquake lights and corona discharges. This also has an effect on groundwater chemistry, which various studies have demonstrated have an effect on animal behavior. This would also mean that these electrical processes would occur at least days before the moment of an earthquake. Laboratory studies indicate that this process might be piezoelectric, meaning the currents might be generated as the rocks are being compressed or stretched. Eugene concedes that this might indeed be the case, but this may be secondary to the metabolic processes or that they might be generated at the same time. One additional curiosity is that it has been found that the distribution of the deepest earthquakes demonstrates a noticeable seasonal variation, which, according to Eugene's hypothesis, might imply the microorganisms are connected to other global cycles, perhaps through the global electric circuit, or perhaps through some other cosmophysical mechanism. So is it possible that we can take this model and examine the possibility of extraterrestrial ultra-deep biospheres? We know that the Moon and Mars have seismic events of uncertain nature. At this stage, there is not enough evidence to determine if the origin of these events is similar to Earth. If they were indeed caused by an ultra-deep biosphere, how might this have developed? It is possible to assume that life itself originated on these bodies, but it is equally possible that life might have been able to colonize them from Earth. One possible scenario might be that microorganisms which have become endospores end up in the ocean and undergo aerolization and lift high up into the atmosphere with a cyclonic updraft and the associated electric current. They might be able to reach the upper ionosphere. From here, they might be able to reach the magnetosphere via the current system connected to the ionosphere. Earth's magnetotail is known to extend all the way to the Moon. This might be the vehicle they need to end up on the surface of the Moon. They might even from the magnetotail be able to end up in the solar wind, which would allow them to escape Earth altogether, and this may allow them to eventually reach other bodies. These endospores would simply wait until the appropriate conditions arise. this may provide a mechanism for the mysterious seasonal emissions of methane on Mars, perhaps connected with some electrical changes in Mars's environment. This is even more plausible if you consider that we now know of the existence of electrotrophic organisms. These electric bacteria are forms of bacteria that directly consume and excrete electrons at different energy potentials, without requiring the metabolization of any sugars or nutrients. This means that in order to survive, they only require electric currents. Since all the rocky bodies in the solar system are surrounded by magnetized current-carrying plasma, and the interaction of the solar wind protons with oxygen-bearing minerals or atmospheric gases constantly producing water. So theoretically, it means that life might exist anywhere. Interestingly, some bodies like asteroids and comets seem to hold a large abundance of complex organic materials, almost identical to high-grade oil shale. This may be an indication of the presence of these microorganisms on the surface, performing ongoing biogenic electrochemical recycling of the rocks. Alternatively, this may reinforce the idea that these bodies represent the debris generated during the planetary catastrophes in the past. Eugene goes on to discuss how the ultra-deep biosphere could play an important role in providing nutrients to the layers above it. There might also be an exchange of genes upwards during the earthquake events, and this may be critical for sustaining diversity and the adaptability of life itself. The ultra-deep biosphere might play a crucial role in providing a seed bank for the whole biosphere, and allow for a fast recovery after a mass extinction and drive the evolutionary innovations, filling the newly created ecological niches with new species. As Eugene has assumed the importance of telluric currents and the integration into the global electric circuit for the ultra-deep biosphere, it seems logical to claim that the electro-autotrophy, so therefore microorganisms that require only current, might represent the second most important type of primary energy and nutrient production. There are two main channels of solar influence: electromagnetic emissions and charged particle emission from the sun through the solar wind. We know that plants have adapted to harness the first and represent the largest reservoir of biomass on the planet. Eugene points out that it would be very strange to assume that no organisms have adapted to utilize the second. The ultra-deep biosphere seems to be the environment where such an adaptation would be most fitting. Therefore, if life evolved on Earth, it might have appeared in the crust first, meaning cracks and crevices of the Earth's crust filled with water and vivified by telluric currents and the associated electrochemistry are the primitive casting molds for the production of life itself. This is supported by the fact that the oldest known fossils of microorganisms are found embedded in microscopic hematite tubes and filaments similar to those of modern hydrothermal vents. One remaining problem for this hypothesis is storing and delivering this energy in order to create an earthquake. One of the biggest problems is that microorganisms at great depth tend to have a much lower metabolism, meaning the estimate of the energy we discussed in part 1 may not be achievable with this number of cells. Eugene proposes 3 possible ways to overcome this. Number 1. The possibility of energy accumulation in the ultra-deep biosphere over an extended period of time. So here the biogenic currents would slowly charge up like a capacitor. It is not clear whether this charge is stored in the cells or whether it is stored in the surrounding rock, which may act like a capacitor. Number 2, that the microorganisms can undergo spikes of significantly increased metabolic rates. And number 3, that there is a serious underestimation of the amount of biomass in the ultra-deep subsurface. Samples taken from deep core drilling do not show an abundance of microorganisms, and this may be partly due to the microbes' inability to survive the rapid transition from where they were extracted under extreme pressure and temperature to the surface. Another big problem is that most bacteria are what are called uncultured. And in a nutshell, this means that no matter what you do with them, they will simply not grow in laboratory conditions. Meaning that the normal way that you would assess how many bacteria there are is to grow them in petri dishes and then to count them under a microscope. But this simply will not work. Another possibility that Eugene suggests is that they may in fact be more exotic types of life that we are not trained to look for. So what are my thoughts on this paper? I have to admit that from the moment I started to read this paper, it struck a chord on many levels. First, the concept that life is far more hardy than we give it credit. The fact that this biosphere plays a crucial role in the total biosphere and that we need to start considering the whole and not the individual parts. I find the evidence compelling to suggest ways that these microbes might survive at these extreme pressures and temperatures, and that we also find evidence of these escape microbes around earthquake faults and volcanoes. I like the idea of linking this with the overall electric circuit. And again, for me, this comes back to viewing this as a whole. I find it intriguing to explore the possibilities that Earth could have and is seeding life onto other bodies, and equally that life was seeded to Earth from a foreign body that happened to pass by. It also presents a different avenue to explore our solar system for life. There are so many aspects that this paper touches on. It is important to realize that at the moment this concept needs more research to verify some of the interesting avenues it throws up. There are still some interesting questions this paper raises which may direct future research. To me, these would include the possible mechanism of delivering the short burst of energy which triggers that earthquake. So again, this comes back to, is it stored in the cells? Is it stored in the surrounding rock? And how is that actually delivered to actually cause that earthquake? Verification of the types and variety of microbes in core samples. So this comes back to, can we find different ways to actually count them? If they are dying, are there different ways that we can look at it? Can we look at what's left over, or is there not much left over after the pressure change? Should we be looking for completely alternative types of life, or should we be taking into account that these are uncultured and therefore find different ways of counting the number of these that there are in these samples? And lastly, the examination for the potential that these endospores exist in our atmosphere or higher up. Again, this should be something that we could test. Can we detect endospores in our atmosphere? And again, can we use that? Can we look at samples that are collected from the moon and identify endospore samples there? In part 3, I want to have a discussion with Eugene about some of these questions and to get into more of the details, as well as discussing the implications that this could have for us here on Earth, for the hunt for extraterrestrial life, and the origins of life itself. As always, be brave, be curious. The truth is waiting for us. Until next time.